Welcome back, students. In this section, I'll be talking about the other water-soluble vitamins. So let us start with niacin. In this section, niacin. What is niacin? It is nicotinic acid. So what is the chemistry, sources in RDA? Chemistry, nicotinic acid is the oxidized form of nicotine. And where is nicotine? Nicotine is present in cigarettes. It is caused due to nicotine. Nicotine is present during smoking and all that. So it is not the same as nicotinic acid. So we cannot interchange nicotine to nicotinic acid. So by smoking, nobody is going to get niacin into their body. Nicotinic acid is entirely different from nicotine. Nicotinic acid is the oxidized form of nicotine, and in the body, niacin is a pyridine derivative. And niacinamide or nicotinamide is an amide derivative of niacin. So nicotinamide has come from a niacin and it is the amide derivative. The coenzyme forms can be synthesized in the body from the amino acid tryptophan. Very important thing, because in the body we can synthesize NAD. 60 milligrams of tryptophan can form 1 milligram of niacin. And the sources are dried yeast, liver, peanuts, whole cereals, legumes, meat, fish. And the RDA is 20 milligrams per day. The RDA is around 20 milligrams per day. So let us look further. The two coenzyme forms of niacin include nicotinamide adenine dinucleotide and nicotinamide adenine dinucleotide phosphate, NAD plus and NADP plus. So what is the difference here? If the adenine is going to have one more phosphate group, then we call it as NADP. Otherwise, it is NAD. So look at the structure: adenine, adenosine, and dinucleotide, and nicotinamide. So this is the whole thing about structure of NAD plus and NADP plus. So what are the functions? It is mainly involved in oxidation, reduction, reaction. It is a redox coenzyme. So can you name any? There are so many of them. All the dehydrogenases, so many dehydrogenases use NAD as the coenzyme and get converted to NADH. Now, what is the difference between flavo, that is a riboflavin derivative coenzyme, that is FMN and FAD, and NAD is that the FMN and FAD are tightly bound. Most of the things are tightly bound to the enzyme. They are not easily removable from the enzyme, hence the word, they are all flavoproteins. FMN containing enzymes or FAD containing enzymes are flavoproteins, for example succinate dehydrogenase, whereas NAD containing enzymes are not, there is no such thing, they are not so tightly bound. It is not a prosthetic group of the enzymes. Niacin derivatives can come out of the protein. They are easily diffusible, and that is the reason where some enzymes use NAD, other enzymes use FAD or FMN. So oxidation, reduction, reaction. Now in this, NAD plus is converted to NADH plus H plus, so it accepts one hydrogen atom and one electron. So one electron has to come here, only then this plus will become negative, plus will get neutralized, plus one proton. So hydrogen atom plus one electron is called as a hydride ion. So how is it? One hydrogen atom, so it is a proton, H plus plus E minus. This is a hydride, and a hydride ion is what it accepts from any substance. When it accepts one hydride ion, what will happen to that H2 that has come out of there? It will go into this may be H plus E minus also. So hydrogen atom, it is not a proton, it is actually a hydrogen atom plus electron. So whenever we have got H2 which is going to come from any substance, H2, one will form full H plus E minus and another will form H plus E minus. So this E minus will go here. So will be accepted by this hydrogen and one electron. Hydride ion will be accepted by NAD plus, whereas this H plus is going to be released into the surroundings. So that's why in all reaction we write it as NAD plus is converted into NADH plus H plus, to emphasize that this electron has also been taken up by NAD. So it accepts one hydrogen atom and one electron, hydride ion. So 2H will form H plus plus E minus. Another H plus is released into the surrounding medium. Now both NAD and NADP are easily dialysable. Remember, the same thing is true for even NADP. NADP plus, NADPH plus H plus. So it's the same thing both for NAD or NADP. Both are easily dialysable. NADH can enter the electron transport chain to produce ATP. We know that already it may be 2.5 ATPs or 3 ATPs. NADPH is used for reductive biosynthesis. So let us look at certain NAD plus dependent enzymes. Now, which are these? So many dehydrogenases are there. Now, some of these which you need to remember include lactate dehydrogenase, pyruvate to lactate. Other is pyruvate dehydrogenase, pyruvate to acetyl-CoA. Other is alpha ketoglutarate dehydrogenase. Alpha Kg, where did it come? It is a part of the TCA cycle. Other is alcohol dehydrogenase, very important because alcohol is getting converted to acetaldehyde. Again, further also NAD is required to convert to acetate. And glyceraldehyde 3-phosphate dehydrogenase. Why? Because it is involved in lipid metabolism for glycerol kinase and all these to act. Glyceraldehyde 3-phosphate is converted to dihydroxyacetone phosphate. So because of all these reasons, these are the important ones which I have listed out here. Lactate dehydrogenase, pyruvate dehydrogenase, alpha ketoglutarate dehydrogenase, alcohol dehydrogenase, glyceraldehyde 3-phosphate dehydrogenase. Any dehydrogenase you take will require NAD+. So NADPH will go next. So which are the, in all those NAD plus requiring, NAD plus was converted to NADH plus H plus. So now let us look at the reactions in which NADPH is produced, where NADPH generating reactions. Why are these reactions important? There are very few of them, and one enzyme deficiency in that can lead to a lot of problems. Already we have talked to you about glucose 6-phosphate dehydrogenase deficiency. NADPH is not produced, but the effects are mainly because of all the different things that are happening is because of the lack of NADPH. So there are two enzymes in the HMP shunt, glucose 6-phosphate dehydrogenase and 6-phosphogluconate dehydrogenase. Remember, the second enzyme can act only when the first enzyme is acted. Glucose 6-phosphate has to be converted to phosphogluconate. Only then it can form phosphogluconate dehydrogenase can act. So if the first enzyme itself is blocked, there is no generation in the second reaction also. So NADPH is not generated by HMP shunt. NADPH is involved in a lot of reductive biosynthesis, lot of effects. It is responsible for maintaining the RBC membrane integrity, etc. So it is not just ATP, there are so many other things which are useful in the body, and those are being done by NADPH. So the two enzymes of HMP shunt. Other than that, there is one more, isocitrate dehydrogenase. Isocitrate dehydrogenase of malic enzyme, they are all minor enzymes. Two major enzymes, glucose 6-phosphate dehydrogenase and 6-phosphogluconate dehydrogenase in the HMP shunt. These are the two important NADPH generating reactions. These are minor, but they take a major role, especially during de novo synthesis. Malic acid, malate is converted to pyruvate. So NADPH is generated in the cytosol so that fatty acid de novo synthesis can take place. So this is happening during the transport of acetyl-CoA from the mitochondria to the cytosol. That is where malic enzyme is going to act and help in the transport of NADPH at the same time. So these are the NADPH generating reactions. Now where is this NADPH utilized? What we call as NADPH utilizing enzymes. So it is mainly in reductive biosynthesis. So synthesis of fatty acids, de novo synthesis of cholesterol, HMG-CoA reductase. Remember, the regulatory enzyme requires NADPH. Synthesis of steroid hormones requires NADPH. All the steroid hormones, corticosteroids, estrogen, progesterone, testosterone, any of this requires NADPH. Phenylalanine hydroxylase, another important enzyme, which can, though it requires tetrahydrobiopterin, there is one isoform which can be utilized, phenylalanine hydroxylase further, and plus in phenylalanine metabolism. Folate reductase also requires NADPH. Now, what will be the causes of deficiency? Can deficiency occur, and what will be the features? Now, the causes for deficiency: dietary deficiency of tryptophan. This is the major factor whenever there is dietary deficiency of tryptophan. And when is tryptophan not taken? Remember, niacin, 60 milligrams of tryptophan can form 1 milligram of niacin. So, deficiency of tryptophan, when does it occur? It occurs in maize eaters. Maize eaters is the most common reason for tryptophan deficiency, because in maize niacin is in a bound form. Okay, so maize eaters are one group of population in which tryptophan is deficient and thereby niacin is deficient. Now, the other group of population which can develop tryptophan deficiency or niacin deficiency is jawar eaters, maize and jawar. Jawar, why does it happen? It is not because niacin is bound there. Jawar contains leucine, which prevents the conversion of niacin to NAD. Leucine prevents the conversion of niacin to NAD. Its active form is conversion is not possible. So more common is jawar. Whatever niacin is there also, it cannot be converted to its active form, and thereby it will lead to all the NAD plus all the enzymes are affected and leading to deficiency syndromes. So both maize and jawar eaters. Now, other than that, it can also be seen in B6 deficiency. B6, vitamin B6, pyridoxal phosphate deficiency. Why? For the conversion of tryptophan to NAD, we require in the pathway one pyridoxal phosphate, the 3-hydroxyethylate. At that level, we require where the bifurcation is taking place, vitamin B6 is required by the kynurenine pathway. So B6 is not there, tryptophan cannot be converted to niacin and can lead to deficiency. Now I have also written isoniazide, INH. What happens in INH? INH actually causes vitamin B6 deficiency. Vitamin B6 deficiency leads to niacin deficiency. Now there is one more disease, Hartnupp's disease, where tryptophan is not absorbed and it is excreted in the urine, and this also can lead to deficiency of niacin. Carcinoid syndrome, again, tryptophan is utilized for the synthesis of serotonin. More and more tryptophan is diverted in carcinoid syndrome for serotonin synthesis. No tryptophan is available for the synthesis of niacin, and thereby it can lead to niacin deficiency. So these are the causes for deficiency. Now, what will be the features? Most of the features are overlapping features which can happen in niacin deficiency. There is no fixed disease. Okay, this may be because of niacin. All the energy forming reactions are affected. So weakness, fatigue, insomnia. It can also lead to different muscle weakness, pain, body pain, etc. Diffuse symptoms, not very clear cut. Okay, and let us now look at vitamin B6, the next vitamin B6. What is the chemistry? There are three forms: pyridoxine, which contains the alcohol group, pyridoxal, which contains the aldehyde group, and pyridoxamine, which contains amine group. So depending upon the group, there are three forms of vitamin B6, pyridoxine, pyridoxal and pyridoxamine. Out of this, all have pyridine ring, and the sources are yeast, cereals, legumes, oilseeds, egg, milk, meat, fish, etc. We have a lot of sources of vitamin B6. The RDA is related to protein intake. It is around 2 milligrams per day. Active form, how is it synthesized? Pyridoxal is important. Pyridoxal is converted to pyridoxal phosphate. Pyridoxamine has to really form pyridoxal, and only then pyridoxal phosphate can come. So minute you have got this, what does it mean? It requires energy. ATP is converted to ADP, and it is a pyridoxal kinase. So pyridoxal phosphate, PLP, is the active form of vitamin B6. PLP, where is it attached? It is attached to the epsilon amino group of the lysine residues in the enzyme. Just like biotin is attached to the epsilon amino group of lysine residues. Lysine residues form a very important residue where all this, the amino group, all these different coenzymes can attach. So it is an important. So with these enzymes, PLP is also attached to the epsilon amino group. What are its function? Now it is a vitamin of amino acid metabolism. So where it is involved in transamination. Transamination form the central reaction of amino acid metabolism. Why? All the amino acids can donate the amino group to alpha ketoglutarate to form glutamate. So because of this forms a central reaction. We also know about ALT, alanine transaminase, and aspartate transaminase, AST, which are even used as liver function test. So ALT, AST, so it is an important vitamin of transamination. In this, remember, in transamination, it is a sort of a ping-pong reaction in which first the amino group is taken up by PLP, and it forms. The enzyme is getting amminated. The pyridoxal phosphate gets converted to pyridoxamine phosphate, and then again it is going to give that amino group to a keto group. The first substrate goes out and the second substrate comes in, and then it gives it to the alpha keto acid, and it forms a glutamate or any other, the newer amino acid. So PLP is involved in what we call as ping-pong reaction, where one substrate comes, it gives something to the enzyme, it goes out, the second substrate comes, it takes it from the enzyme, and it is converted to a new product. So this is transamination. It is also involved in decarboxylation of amino acid. So you are found out in biogenic amines, tyrosine to tyramine, dopa to dopamine, histidine to histamine. So in all these biogenic amines, there is decarboxylation that is taking place. The enzymes are decarboxylases and they require pyridoxal phosphate. Pyridoxal phosphate is also involved in synthesis of heme. The first reaction, succinyl-CoA plus glycine to form delta amino levulinic acid, requires pyridoxal phosphate. It is also involved in the synthesis of niacin coenzyme NAD from tryptophan. So in B6 deficiency, we will even have NAD deficiency. It is also involved in what are called as trans-sulfuration reactions, because of phosphoadenosyl phosphosulfate. This is required during the cysteine metabolism, and further PAPS is formed. So PAPS is involved in transsulfuration reaction, which is required for formation of sulfolipids or sulfoproteins. So in all this, vitamin B6 is required. It is involved in deamination reactions also. It is involved. And one important connection, all these were with amino acid metabolisms. One important connection is with carbohydrate metabolism is glycogen phosphorylase. This enzyme, though it is with glycogen metabolism, this phosphate is stabilized by pyridoxal phosphate. It is also involved in the synthesis of sphingolipids and in myelin sheath. So what will be the deficiency manifestation? What are the causes of deficiency? Mainly one important cause is isoniazid, INH, which is an anti-tubercular drug. Always B6 is supplemented along with the tuberculosis therapy. Cycloserine, which is used in cancer therapy. Oral contraceptives can also lead to vitamin B6 deficiency. Penicillamine, which is used in Wilson's disease and rheumatoid arthritis. Ethanol. And the deficiency manifestations include neurological manifestations, dermatological manifestations, and hematological manifestations. So let us look at that. Neurological, it will cause peripheral neuritis. It will cause depression, irritability, nervousness, and mental confusion. Mainly these are because of lack of serotonin, GABA, norepinephrine, epinephrine due to demyelination. Dermatological leads to lack of synthesis of niacin. So niacin deficiency symptoms, pellagra. Now in niacin, I said it can lead to all different energy, weakness and all. But more importantly, niacin is associated with pellagra. What is pellagra? 3 Ds. Diarrhea, dementia and dermatitis, and all this is, and finally death. And pellagra niacin deficiency causes pellagra. And in pellagra, what is important are the 3 Ds. Okay, diarrhea, dementia, dermatitis and death. And this is finally death. All this is because of the energy content. This is not forming ATP, so dermatitis. It is not forming diarrhea because of again the mucosal cells of the GI tract are affected, and dementia also because of the various reasons energy is required by the brain. Whereas in vitamin B6 deficiency, it leads to hematological, dermatological effects. So dermatological is mainly because of pellagra in B6 deficiency. The hematological defects will be hypochromic microcytic anemia due to lack of heme synthesis. So how is B6 assessed? There is what is called as tryptophan load test. A load of tryptophan is given and urine is checked for the excretion of xanthurenic acid. In B6 deficiency, there is increased excretion of xanthurenic acid. These are all outdated tests. There are many more tests which are now available directly. So with this, we come to the end of two important vitamins, niacin and vitamin B6 or pyridoxal phosphate. Please remember that niacin deficiency leads to pellagra, and pyridoxal phosphate or vitamin B6 deficiency can lead to different conditions which are hematologically hypochromic microcytic anemia, dermatologically it is because of pellagra which is caused because of B6 can lead to niacin, niacin can lead to pellagra, and finally neurological because of its effects on myelination. Peripheral neuritis is very common in vitamin B6 deficiency. So with this we come to the end of two important vitamins, niacin and vitamin B6. Thank you.